one of the great lessons I learned in this project was not only that we are speaking different languages, even if we are using the same language like English, we don't necessarily understand each other. And that's especially true in these multi-collaborative projects with a lot of different expertises as background. And it was really surprising to me to see how much struggle that means, just to know what we are talking about. So one of the 3 pillars of Driver B is innovation. And everything was fine. We agreed on innovation as long as I, again, caused trouble. And I caused trouble by asking, what do you mean? What is innovation? That really was the beginning of a long discussion. And I tried to find some help in the center of innovation, Silicon Valley. So I looked at cell phones because we all have cell phones and we know what it means, and we had the old cell phones with the keys that usually didn't work after 2 years, and then there was the real innovation coming. That was the touch screen that enabled a lot of different new things. So everyone agreed that was innovation. So we have our cell phones now, and then came all these variations, modifications, improvements, like bigger screens. So we called that, it's novel, it's new. We didn't have that before, but it's not an innovation step. And then there was a discussion about lots of different things are useful. So new apps are useful, and they can be useful without being innovation or even without being really novel, like new apps. So what does this all mean now for us, for the definition of innovation in this context that we are discussing here? So translating the innovation to the scientific context, it's quite difficult to just use these very known things like new class and so on. So we decided to use the definition that really matters to patients and clinicians, and that is having a new antibiotic that does not show cross-resistance to existing drugs. We have heard yesterday in the opening speech how important that can be in lots of different clinical situations. This lack of cross-resistance can best be achieved if you have a new class, could be a new chemical or functional class, new targets, new binding sites, new mode of action. I did not preclude, because we had all exclude, because we had a lot of discussions. It could happen that a modification of an existing class is so profound that it also prevents cross-resistance. Improved antibiotics of existing classes usually reduce very specific class-specific resistance mechanisms. So there are improvements, and for sure very helpful for certain situations. And other improved features, of course, are useful for patients. So now, knowing what scientific definition is does not mean that everyone agrees. So we are still, I know that there are a lot of different interpretations, definitions are around, and some are even including everything that I just showed as innovation. So it's just very important to define what we mean when we say innovation. Yeah, so who is now in this business of innovation? Who is doing antibacterial drug discovery? Of course, SMEs, small companies that are really focusing on antibacterial drugs are around. We have estimates. We don't, of course, don't know exactly the number of SMEs in the world, but it's quite a good number of SMEs that are focusing on antibacterial drugs. Other players are as important, but we don't know how many of them are really focusing on this area. We have really no idea how many academic institutions, how many other publicly funded research institutions, how many non-profit institutions or public-private partnerships are out there. I could just give you an estimate, but it doesn't make sense because I know it's just a number that's probably not real. There are other players, and we all know that the big pharmaceutical companies seem partly to be interested again. And there are companies that we know are active. It's Novartis, it's Roche and Genentech, it's Sanofi, it's MedImmune for the biologics, GSK and Merck. But there are also other large companies that are usually not included in these global pharmaceutical corporations that are quite active from the Asian region and Japan and in India, like Shionogi and Wockhardt. So we shouldn't forget this kind of companies. We don't know. I mean, there are numbers out there, but I just say now we don't really know how much public and philanthropic funding is used, really dedicated to antibacterial drug discovery. We know approximately how much is spent for AMR, or the field of therapeutics, but we don't know how much of it is drug discovery and preclinical development. So that would be worth getting a better idea of who is really doing this work. Another important task of DRIVE-AB was looking at what is happening currently. So what are the pipelines? What is in clinical development and what is in preclinical development? Because that's really very important for estimating the need of rewards or even knowing which drug would be the right one for an award. So we looked on the clinical pipelines. We started this work, and then the WHO pipeline analysis project was started, and this is partly based on this work that will be published very soon, I guess. So please don't look at individual names, individual drugs. That's not what I want to show you, just at this color scheme. And in this slide, you just see one class of antibiotics. And these are the beta-lactam class, either in combination with beta-lactamase inhibitors or not. So one class of antibiotics that's now in clinical development. And I've listed here only the WHO critical pathogens. So here it's all carbapenem-resistant pathogens, Enterobacteria, Pseudomonas, and Acinetobacter. Green means there's likely clinical activity or usefulness. Yellow is we just don't know or it's unclear, and green and white is most likely no activity or insufficient activity. And you can see there's really very little other than spotted activity, like part of the enterobacteria would be covered because there are different beta-lactamases. And as I said before, these modified classes are usually addressing very specific, class-specific resistance mechanisms. So if you have other resistance mechanisms, then it would not work. The message in general is that despite a lot of drugs in the clinical pipelines, there is still not really much around which addresses the critical priority pathogens. And if, then not completely and only partly. So the lesson from this slide is we have a pipeline, and this pipeline addresses specific solutions for specific patients, for specific situations, in specific regions. This is great and useful for exactly these situations, but it does not really address the public health need. Of course, this is just one class of antibiotics, and we have more. This is the rest of the pipeline, the clinical pipeline. So here's phase 3, so they are expected to come quite soon. And you can see a lot of white, much more white than green. Here, additionally, here I have the blue column, and that means gram-positive activity. So clearly, these antibiotics have been focused on gram-positive pathogens. And that's understandable because that is still the consequence of the MRSA bad situation 15 years ago. So everyone was focusing on MRSA, and I remember investors at that time, they said, we don't even listen to a company if it doesn't have an MRSA drug. So now we get the result of this prior situation, and a lot of different drugs that are probably active against gram-positive bacteria. And relatively still not much innovation in the sense that I defined before. So the red ones here are the new classes, new targets, new mode of action. So the clinical pipelines, you could call them, they are filled, but not necessarily what we need right now. So what about the preclinical pipelines? So these are the drugs that need much longer time to be available to patients. Is it the same situation, or do we see a change in focus? So we are thankful to Capex and Kevin Ortison, who gave us insight into the Capex submissions of the last year. These were a lot of projects that we could analyze. And of course, it's not a total picture of what's happening in the world, because it was the first year of these funding cycles. But I still think it gives us quite a good indication and a good idea what's happening in the much earlier stages. So again, you don't need to look at details. The first column, red, you know, I'm a microbiologist, and red always means gram-negative. Blue means gram-positive. So red means these are the projects that are focusing on gram-negative pathogens. And these are the ones with new classes. So these are the ones that we could call innovative approaches. The blue ones are the new ones against gram-positive pathogens. So most of these projects were coming from small companies, few from academics, large companies, and also non-profits. So what do we recommend for push mechanisms? We need to broaden the base. And we also know that discovery grants improve the entry rate into the preclinical phase. The more discovery projects we have, the better the chance of getting something through this cascade. But it's not enough to just give more money, more push funding. In general, we also need to prioritize some of them for exactly the main problems that we have. How do we do that? We need to increase, coordinate, and optimize our resources. That's absolutely necessary to avoid duplications. So addressing drug discovery and early development hurdles, addressing them in a scientific but also financial way. And the Pew Trust has done some work to really show what the hurdles are. And of course, we need a global coordination hub. Additional funding is still necessary in addition to increasing, coordinating, and optimizing resources. The difference between push and pull is that for all projects that are pushed, we have a much higher attrition rate, and for the pull, we have a certainty, relative certainty what we get. So for the research, this is applied research around the topic of resistance. These are mostly broad topics, but still we need to coordinate and increase funding and optimize the resources. So we have to address the most difficult scientific questions. It's a little bit different for drug discovery because that's not in general resistance. It's really getting a drug as a goal. So that will be still broad because we don't know exactly how a drug discovery project will turn out, but also include priority topics. And this affects especially academic, non-profit institutions. They need a little bit more than just funding and prioritization. They need also some support in knowledge and partnerships. SMEs, we need to increase the focus on expertise and experience because we have lost a lot during the last decades. Drug development is more now focusing on priority pathogens, but also on target product profiles. So we need to define from a public health perspective what is really needed. In this case, especially in the later stages of clinical development, if there are push funding, then there needs also to be these conditions for sustainable use and equitable access conditions to apply. So in general, the earlier and research and drug discovery projects are, the broader the scope and the closer they come to the clinic, the more we should focus on priorities. So I'm almost at the end. We had this discussion started for the pipeline coordination about 1.5 years ago. And this idea was focused on public health outcome-driven coordination of pipeline activities. So that means identifying gaps in the current pipelines and addressing these gaps. So these early ideas of a global collaboration hub on AMR research and development and the integrated public health-driven R&D organization, they already happened. So that's a typical example also about our situation in Drive B. We had ideas, we were developing it, we were discussing it, and in the meantime, the situation has changed and politicians were much faster than we could be in our project. So that means, as you have heard yesterday, the G20 under the German leadership has already initiated and tries to really get this done and implement that. So maybe we should just not try in Driver B to implement things. We are just waiting and they implement themselves. So CardP, we have heard yesterday, also we are successful. Just my last two slides on these two initiatives. So this global collaboration hub, WHO has done a lot of work already as a really important basis, like the priority pathogen list and the pipeline analysis, and hopefully they will also do target product profiles. So identifying gaps, align existing funding, coordinate new funding, coordinate new push but also pull incentives, so it's along the whole pipeline, and also define policies for sustainable use and access. So this all should inform public-private philanthropic decision-making, increase efficiency, and hopefully will lead to a new antibiotic. It should be global, really everyone, all players should be integrated and work on this. So finally, the last, that's the CardP model. The very special thing about CardP is that it has, from the beginning to the end, a public health focus. So whatever they do is focused on public health, and whatever phase they're in, so whatever is the point of entry to patient delivery, they're integrating affordable, equitable, and sustainable use provisions. That's their whole thinking. So they have in-house scientific R&D, otherwise they are working like a virtual company, and they have a global perspective based on partnerships. So I think, I just hope that it will be successful, but the start was quite good. So as 2 days ago, it was announced that they got some money. So this is the end now, and a very short summary of these push mechanisms from grants. Of course, there are so many more details that we worked on. Push mechanisms, but also this pipeline coordinator, and I'm sure there will be much more details coming up in the next time. Thank you.